O.Nureki from Japan, who is one of the gurus in the structural biology of Crispr Cas system. And without any further delay, he'll talk about how you can convert knowledge about structure into something very, very useful.
Thank you very much for introduction, and I first thank to the organizers, especially the Dr. Zakian, to invite me to this very interesting symposium. Actually, it is the first time for me to come to the Russia, and I'm so excited. So I'd like to talk about the molecular mechanism of Crispr Cas and the structure-based development of the genome editing tool towards medical application. So I would like to introduce some history of the CRISPR. So 1987, Dr. Ishino in Kyushu University read the sequence in the downstream of the E. coli alkaline phosphatase enzyme, and he found that the very interesting structure that the some 29 nucleotides directly repeat, and after that you have a different distinct 32 base spacers. So, but these are repeated by 5 times. But he said that these sequences have no homology to the some prokaryotic gene, and so maybe this is from the some extracellular gene. And but he said that he could not understand the biological significance of the sequences, and he stopped the investigation. So, but later, the investigators in the Europe and the United States uncovered that this is related to the acquired immune system of bacteria. So, bacteria is attacked by the virus called phage. So, phage introduced the external foreign DNA to hijack the bacteria. But bacteria had the Cas1 and Cas2 enzymes which cleave this fragment of this foreign DNA and integrate it into the some region in the bacterial genome called Crispr RNA. So this is found by the Dr. Ishino, and after then this spacer region on this directed repeat region transcribed to produce the non-coding RNA called CRISPR RNA, as you know. And the bacteria also prepare the another non-coding RNA called tracer RNA, and this tracer RNA and CRISPR RNA form the complex, and further form the complex with the immune protein Cas9. Then this complex waiting for the coming of the external foreign DNA and cleave it to stop the hijack by the phage. Sorry. So in 2012, Jennifer Donner in the UC Berkeley and Emmanuel Charpentier at the time in the Vienna University reported that the Cas9 is a probable dual RNA guided DNA nuclease acting in the adaptive bacterial immunity. So that is the this is Cas9, and this is the CRISPR RNA. This is trace RNA. So CRISPR RNA unwind the double-strand DNA. Then the LAMC and HNH nuclease domain in the Cas9 cleave the single-stranded region of this non-target and target strand respectively. And but the bacteria should not cleave its own DNA. So bacteria recognize this protospacer adjacent motif PAM sequence to recognize, to discriminate self and non-self. And so this pump sequence is located on the non-target strand. And they conjugate the CRISPR RNA and the trace RNA with the artificial tetraloop to form the single guide RNA. So you can perform the gene editing using a single guide RNA. Then, half a year later, Fandon in the MIT Broad Institute reported the first genome editing in the mouse and the human cells. So, in eukaryote had a nucleus, and in the nucleus you have a chromosome, and the chromosome are folded to the chromatin, and you have a double-strand DNA. So Cas9 is guided by the guide RNA to bind to the specific sequence in the genome here, and make a double-strand break. Then cell autonomously repair this double-strand break, and in two ways. First is non-homologous enjoining, so join the two cleaved DNA, but the cleaved DNA is unstable, and you have a deletion or insertion. So if you have a gene here, the gene is knocked out. The other way is the homologous directed recombination, homologous recombination. So you have a homologous DNA, and the fragment is integrated, or you have a knock-in here. But the rate is, this is 99%, this is less than 1%. But this technique can be used for the gene therapy, and also you have two strategies. First is ex vivo gene therapy. You can extract the cells and you perform the genome editing and to the cell in vitro. And then if you have a successfully genome edited cells, you can transplant the cell to the back to the body. So CAR-T is the famous technology using this gene therapy. The other strategy is in vivo gene therapy. So you can package the Cas9 and the guide RNA and the template DNA into a virus, and you can make intravenous injection. Then the virus delivers to the liver or brain, and you can perform the genome editing in the body in vivo. This therapy is very speedy and efficient and very clean because you target the specific gene, designed gene, and you have no problem of donor and transplantation rejection because you edit your cells. So we started the CRISPR project since 2012, but before with the collaboration with this Fanzan, but before that we met with Fanzan in 2009, and at the time he was the graduate student in the Stanford University under the supervision of the Professor Carl Daisaros. And we saw the structure of this channel rhopsin, which is very useful for the neuroscience. And if upon the right perception, this channel rhopsin becomes the channel and conduct the cations to activate some specific neurons. But after that, Fan become PI in the Harvard and MIT and start the genome editing project. And he recommended us to collaborate about this project. And so this assistant professor, Dr. Nishimasu, mainly managed this project. So we saw the first structure of the Streptococcus pyogenes SP-Cas9 complex with this yellow target DNA and red single guide RNA. And so in its structure, you have two lobes. The first lobe is the recognize the nucleic acid. So this is called as the recognition of red lobe. The other lobe is contain this HS domain and RABC nuclease domains. So this lobe is called as nuclease lobe. And this red lobe and nucleus lobe have a central channel at the interface, and this interface accommodates the RNA DNA duplex and the rest of the guide RNA. But cleavage side of the target strand here, but we have cathodic residues of the AGN domain here. They are separated. So this means that this structure represents the inactive or resting state. The active side of the rhabs domain is here, so non-target strand should be around here. So, and this is nucleic acid bound to the Cas9. So this yellow represents the target DNA, and this blue light and blue and this red represent the guide RNA. So here you have a separation of the target strand and the non-target strand. So in this structure here, target strand and non-target strand are separated, and this the first 10 to 12 nucleotides in the guide RNA, this blue light guide RNA, is called the seed sequence, which is very important to separate the target strand and non-target strand. So Cas9 should strictly recognize this seed sequence in the guide RNA. So we found that this seed sequence is recognized by the many arginine residues protruded from the long alpha helix called as the bridge helix. And this bridge helix connect the red lobe and nuc lobe, and but these arginine residues recognize the phosphate backbone of the seed sequence in the guide RNA. So we mutated these arginine residues to the alanine and analyze the knockout activity in hex cells. And we found that this arginine 70 and arginine 74 is very important for the knockout activity by the Cas9. And you see that the arginine 70 had the 3 hydrogen bonding or electrostatic interaction with the phosphate backbone of the seed sequence. And also this arginine 74 provides the 3 hydrogen bonding or electrostatic interaction with the phosphate backbone of the guide RNA. So these arginines are very important for the recognition of the seed sequence in the guide RNA and important for the separation of the target and non-target strand of the genome DNA. We have another important point that here this target strand and non-targets are separated, and actually this phosphate group had the flipped out configuration to stabilize the wound conformation of the double-strand DNA. So in a structure, this flipped out phosphate here is recognized by 3 hydrogen interaction by this loop called phosphate loop group. Not only in the SP Cas9 but also the Cas9 from the Staphylococcus aureus. So this structure stabilizes the unwound conformation of the double-strand DNA. Finally, the PAM sequence, PAM, should be here on the non-target strand, and this yellow C-terminal domain called as PAM interactive domain may recognize the PAM sequence. And a half year later of our paper, the Martin Jinek in the Zurich University reported the SP Cas9 complex with the target, the guide RNA, and the double-stranded target, the genome DNA. So it includes the non-target strand, so it has the PAM sequence here. The PAM sequence for the SP Cas9 is 2 guanine and GG. So he revealed that these 2 guanines are recognized by the 2 extending arginine residues by the totally 4 hydrogen bond interaction. So 2 guanines are very strictly recognized by the Cas9, SP Cas9. So this is the structure solved by the Jennifer Dana group and published a week before our third paper. And this is a complex structure of the SP Cas9. These are the same SP Cas9, but they are nucleobe had the same conformation, but the regulobe had a completely different orientation. So they think that this apo form had the inhibited form for awaiting for the coming of the guide RNA. And they build some movie that this is the nucleomane, this is the red lobe, and upon binding with the nucleic acid, the red lobe change its orientation largely and make a central channel which accommodate the DNA duplex and the rest of the guide RNA. But we are wondering if the Cas9 had such a inhibited form. So we perform this high-speed atomic force microscopy. So in a atomic force microscopy, there's a thin needle in the cantilever touch the surface of the molecule, and using this laser, you can visualize the touching, convert the touching to the visualization. So you can visualize molecule using this AFN. But you should fix the sample on this plate. So and also the procedure is very slow. So we speed up every procedures, and we can see the dynamics of the molecule. So we put the liposome, and the liposome glow to the membrane bilayer. So we can loosely fix the sample on the lipid bilayer, and then, for example, you can see that this is actin, this is myosin. So myosin is working on the actin. You can see such a dynamics of the protein. So we can utilize this technique to the Cas9. So in this apo form, here, I have a movie here. Could you? Sorry, it's done. But we would like to examine if the Cas9 had the apo form, had the inhibited form, but in this movie it shows that the red lobe and the loop lobe are fluctuating and has no static or inhibited form. But in a complex with the guide RNA, it had a static complex. It's a sub-effective complex. So then we add the DNA to the Cas9. This is DNA, this is the Cas9, and you have a H domain here. Now H domain migrated to access to the cleavage point and create a DNA. So you have a cleavage of the DNA here. So this is the first visualization of the DNA cleavage by the Cas9. So, but for the genome editing, SP-Cas9 had mainly two problems. The first is SP-Cas9 is too large. It contains 1368 residues. It causes the low efficiency of packaging into the viral vectors to deliver to the somatic tissues. The second problem is that SP-Cas9 very strictly recognize the PAM GG sequence, which called the limitation of the target selection. So we'd like to solve this problem based on a structure. First is mini Cas9. So SP-Cas9 is very large, but Cas9 from the Staphylococcus aureus SA-Cas9 is smaller by the more than 300 residues, and in the DNA level, SP-Cas9 had 1 kilobase, 4 kilobase, but the SP-Cas9 had 3 kilobase. And but if the Cas9 is smaller, the Cas9 recognizes the longer PAM sequence. So because in the case of the SP-Cas9, it recognizes GG, 2 nucleotides, but SP-Cas9 recognizes G, purine, purine T, so 4 nucleotides as a PAM sequence. And the guide RNA for each Cas9 is so different in the sequence and the secondary structure. But we saw the structure of the SP-Cas9 complex with the guide RNA and the target DNA. Here, you can see that we have some truncation of this domain and here, so we have a more smart Cas9. This is the HN domain protruded by this long arrow helix, and this is the LAPC domain. And we have here, we have a long PAM sequence, G purine purine T. So now we know the mechanism how this ACCAS9 recognizes the 4 nucleotides as a PAM. So the first G is recognized again by this extending arginine residues, but the second and the third, the purine residues, are recognized at the nitrogen atom at the N7 position by the 2 aspargine and the 1 arginine residue via water molecule. So these are represent some fuzzy recognition. And the terminal T is recognized by this bending arginine residues. So we mutated these arginine and aspargine residues to examine the knockout activity in hex cells. So 1 point mutation reduced at most half of the in the knockout activity, but the double mutants completely damaged the knockout activity. This means that recognition of the PAM is not only to discriminate self-unknown cells, but also to trigger the unwinding of the double-strand DNA. So this is called as the helicase activity of Cas9, which is very important for the double-strand break. And so this is SA-Cas9, this is SP-Cas9. So SP-Cas9 has an extracellular region here. So if we truncate these regions, then we have a small smart SA-Cas9. But we search for the more compact Cas9, and that is the Campylobacter jejuni Cas9, CJ-Cas9, which has only 984 residues. We saw the structure of the CJ-Cas9 complex with the guide RNA in red and double-strand DNA in black here. So we have a longer PAM sequence, but this PAM sequence is recognized not only in the non-target strand, but also the target strand, especially this G, guanine, is recognized by this arginine. So both strands of the PAM duplex is recognized by this CJ-Cas9. And so we found that the CJ-Cas9 recognized the 5 nucleotides as a PAM sequence, V, A or 0C or AZ or CT or AC. So 5 nucleotides. So then CJ-Cas9 recognized both strands of the PAM duplex. And so we have some repertoire of the Cas9 structure from the smaller one to the largest one. The last one is this first Novicilla Cas9, FN Cas9, and in this case you have a large wedge domain in yellow. But if the wedge domain is shrinked, then you have a SP Cas9, and you have a truncation of this domain, then you have a smart SP Cas9. But in this case, wedge domain is enlarged. Then the wedge domain is shrinked, you have a minimal CJ-Cas9. The problem of the CJ-Cas9 is that it has a very weak activity in vivo, so you cannot easily use this in the genome editing. Actually, we have only 2 papers that is report the in vivo genome editing by CJ-Cas9. So we based on the structure, we incorporated some many mutations to activate this CJ-Cas9, and this is wild type. Wild type had a higher activity in some PAM sequence, but a very slow activity against for the some PAM sequence. But the mutant has a higher activity for all the PAM sequence. So this activated mutant is very useful for the in vivo genome editing. So I would like to change to the PAM simplification. We would like to make a Cas9 mutant that recognizes single G as a PAM, because this PAM simplification is not only useful to perform genome editing more freely, but also allow you to precisely perform the base editing and transcriptional regulation. These are the non-cleaving genome editing. So if you inactivate the Cas9 by the incorporation to the catalytic residue of the lysine and H domain, you have a dCas9. And if you fuse the epigenetic regulator to the dCas9, you can switch on off the downstream gene. This is the transcriptional activator and repressor. Or you can, if you fuse the deaminase, which converts C to T or A to G, then you could do base editing. In this technique, the PAM-flex Cas9 that recognizes simple PAM is very important to precisely activate the downstream gene or precisely change the base in the base editing. So our aim is to change the GZ PAM to GPAM in the SP Cas9. So, but in the structure, as I said, the two Gs are recognized two adenine very strictly. So we change the second adenine to recognize second guanine to adenine. Then the PAM is now single G. But in this case, the helicase activity is weakened. So we introduced the newly introduced the two adenine residues to recognize the phosphate group in non-base specific manner. Then this recognition is enhance the helicase activity and cover the lack of the specific interaction between this guanine and the adenine. So we incorporated these residues based on the structure, and we purify the mutants. So if we change the second adenine to adenine, we lost the cleavage activity in vitro. But if we add the another adenine residues, then we have a recovery of the cleavage activity. So if we combine these mutants, ARRR mutants, recover the helicase activity and cleavage activity. But in the time course, this mutant has a weaker activity as compared to the wild type. So we see the structure, and here, if we have an adenine here, we have much space here. So if we change this adenine to the valine, then valine occupies this space and stabilize the conformation. And if we change this glutamate to the phenylalanine, this phenylalanine provides the fundamental interaction with the ribose moiety of this G2. So based on these strategies, we incorporate another mutation. So if I add this valine, then activity is increased. If we add this phenylalanine mutation, increased. And if we change this adenine to valine to occupy the space, then we have a very high activity mutant that recognizes single G as a PAM. So we named this mutant and this mutant as NG1 and NG2. So this is wild type. Wild type recognizes only GG PAM, not GA, GT, GC PAM. But our mutants recognize not only the GG but also GA, GT, GC PAM. And in the time course, our mutants not only recognize GG but also GT, GA, GC PAM. And PAM discovery revealed that the wild type recognize GG, but our mutant recognize G, single G. And we compare the activity with the CPF arm, the new type 5 CRISPR. Then this is a CPF arm activity, and this purple represents our mutants. So our mutants has higher activity than the CPF arm. So our mutant is useful for the genome editing. And we saw the structure of the mutants, and these adenine residues recognize a phosphate backbone of the PAM duplex, and these valine and phenylalanine provide a fundamental interaction with the ribose moiety to enhance the helicase activity. But we submitted the paper to Science in January 2nd, and then in February, the David Liu in the MIT and Harvard suddenly published their paper in Nature article, the XCAS9, and that is SPCAS9 mutant that recognize the single GPAM. We are so surprised, but they made these mutants by incorporation randomized mutation to the entire gene and screen the single gene recognizing Cas9 using this phage system. But when we map their mutation, XCAS9 mutation in the structure, you see that most of the mutants are apart from the interface with the nucleic acid. Only these mutants have a meaning. So this glutamate is replaced by the valine. So this glutamate supports the orientation of the second adenine, but if this is changed to valine, this adenine has a flexible, and XCAS9 has a more flexible recognition as a PAM for the PAM. But we purified wild type, our mutant, and XCAS9 and compare the in vitro activity. So mutant recognize GA, GT, GG, GC, PAM, but XCAS9 did not recognize GA, GT, GC, and GG recognition is also, even the GG recognition is weaker. So in a what time course, our mutants recognize all the PAM and GPAM, but the XCAS9 recognize only GGPAM. And a higher dose condition, XCAS9 weakly recognize this GTPAM, so we perform the in vivo genome editing using hexel, and this is a wild type. Wild type in two genes. Wild type recognize GG PAM, but our mutants recognize GA, GT, GG, GC. GC is a little weaker, but all the energy PAM in vivo. But XCAS9 only recognize GG PAM and very weak. So in the very recent result, revealed that the XCAS9 has almost no in vivo genome editing activity. But our mutants has NG1 and NG2 has a very high activity, not only in the NHEJ but also the HDR. So we performed the genome editing in vivo in the many genes, and we found that our mutants has a higher activity, much higher activity than the XCAS9 for the NG-PAM. And we analyzed the off-target activity. So this is wild type, this is a mutant. Our mutant has a higher off-target activity, but incorporation of some mutation to this, that is an eCas9 mutation, then our mutant has less off-target activity as compared to wild type. So we inactivate our mutant by the dCas9, and we add the deaminase, and and we perform the base editing analysis. So wild type recognize GG and edit the base, but our mutants recognize GA, GT, GG, GCPAM to perform the genome base editing. But XCAS9 has a very weak base editing activity. So you have maybe. So I would like to introduce one more new Cas9, new Cas, that is the type 5 CRISPR. So Cas9 belongs to type 2, CPU farm belongs to type 5. These Cas are very different. Cas9 has two nuclease domain, HNH and RABC domain, but the CPU farm only has the RABC domain. And also they have different properties. So Cas9 utilizes CRISPR RNA and trace RNA, but the CPF1 only uses CRISPR RNA. And Cas9 recognizes GG-rich PAM, but the CPF1 recognizes pyrimidine-rich or TTT PAM. And the Cas9 makes a blunt end near the PAM sequence, when CPF1 produces the sticky end apart from the PAM sequence. So we construct the CPF1 complex and solve the structure. So this is the CPF structure. The overall structure is very different from the Cas9. But this is nucleic acid. This is red to represent the guide RNA. This is the 5'. This is the 3', and this is the target DNA. So this is DNA duplex, and but similar to the Cas9, the sequence of the guide RNA is recognized by this histidine or arginine or lysine residue from this wedge domain, not from the bridge helix. And also the flipped out phosphate group is recognized by this phosphate group, but it is in the also in the wedge domain. And we designed it so that the DNA and RNA have a 20 base pair, 24 base pair, but at the 20 base pair, this tryptophan interact between the RNA, guide RNA, and the target DNA, and target DNA wind toward the this RAMC domain or this newly found nuc domain. So we mutated this tryptophan. It lost the mutant lost the genome editing activity. So this is PAM recognition. The TTT PAM had the TA PAM duplex. So H-rich DNA is easily changed its conformation, and actually this PAM duplex is recognized with 3 domains: PI, wedge, and back-one domain, and in this case, the PAM duplex is twisted, and when DNA is twisted, the minor groove is widened. Then this lysine residue enter into the minor groove and recognize the minor groove of the PAM duplex. So in our case, this lysine 607 recognize this base, and this base, and this ribose moiety. And so this is very different from the Cas9. Cas9 recognizes the PAM duplex to the major groove side, but the CPM recognizes the minor groove side of the PAM duplex. So the nucleus domain, we have a RAMC domain and a new nuc domain. And if we change this kinetic residue in the RAMC domain, when the genome editing activity is lost. But when we change these conserved residues, only the mutation of this arginine damage the genome editing activity. And nowadays, similar Cas 12b structure of the Cas 12b was reported. And in the structure, the DNA is wound and cleaved by this RAMC domain. And this arginine guide this target DNA toward the RAMC domain. So this is Cas9, this CPF1 overall structure is so different, but the H domain is inserted in the former part of the RAMC domain and cleaved here. So we have blunt end here near the PAM sequence. But in the case of the CPF1, this nuc domain is inserted into the RAMC domain in the latter part. So it cleaves, and this target strand is cleaved by the RAMC domain. So you have a sticky end. It's apart from the PAM sequence. So we have some mutant that recognize a different PAM sequence, not TTT, but TAT or TYC. And we saw that the structure, actually this wild type did not recognize TAT, but this RVR mutant recognize TAT. Or wild type did not recognize TCC, but this RVR mutant recognize TCC here. So we saw the structure of the RVR and RR mutants of CPF1 in a complex with the guide RNA and target DNA. and we crystallize Dr. Yamano some of the structure, and you can see that in the RVR mutants, this thiamine that form base pair with this adenine is strictly recognized as the newly incorporated adenine and this racine residue. In the other mutants, this palm is TCC, but the pair guanine residues are recognized strictly by this newly incorporated racine and adenine residues. So this is the reason why these mutants are recognized in different palm sequence. So maybe I should maybe.
Okay, so I think you have only need to go.
Yeah.
It's fine.
Okay.
Okay.
Yeah. It may I'd like to apply our technology to the treatment of genetic disease, that is hemophilia. So you cannot stop the bleeding, and it's caused by the defect of the coagulation factor 8 and factor 9 in the X chromosome. So it only affects the boy. So once per 5,000 boys are affected by this genetic disease. If you have a defect in the factor A, you have a hemophilia A, and if you have a defect in the F factor 9, you have a hemophilia B. Then if you treat this genetic disease, you should intravenous injection to a baby 2 or 3 times per week. But this injection is very painful, so baby is crying. But if you can treat this disease by a single injection, baby is happy. Baby is happy. So we would like to make a hemophilia mouse by the genome editing. So we perform genome editing for the embryo. We can easily make a hemophilia mouse. But the germline genome editing cannot be applied to humans at present because of the ethical and the safety concerns. So we should deliver the Cas9 and guide DNA and template DNA to the somatic tissue cells to treat this genetic disease. But the embryo has only single cell, but the liver that produces the coagulation factor is composed of the 1 trillion cells. So we should deliver Cas9 and guide DNA to the 1 trillion cells. It seems very difficult, but we try. We use a smart Cas9 and we make a single intravenous injection to the tail, and after 16 weeks, the factor 9 is reduced to 0, and we could succeed in making a hemophilia mouse by a single injection. To treat this hemophilia mouse, we have a long intron between X1 and X2 of the factor 9. So we cut here that Cas9, and if we are lucky, we have a homologous recombination. But we are unlucky, but we can insert the factor exon 2 to 8 to this cleavage site, and we will have a complete gene here. So after single intravenous injection, and 6 to 8 weeks after, factor 8 is recovered, and the coagulation time is reduced. So we could treat the hemophilia with a single intravenous injection. So we built a venture called EditGene. Laboratory is located in Boston, but we have such a member, and they are using the DCAS9 fused with this some epigenetic regulator to regulate the expression of the downstream gene. This is called the Crispr Genome system. And our aim in our laboratory is to make a super Cas9, and that has a high fidelity with less off-target and more freely attack the target, the genome sequence by the recognition of the simple PAM, and size is smaller, which is very useful for the introduction to the somatic tissues. So finally, I would like to introduce my Dr. Nishimasu. He's the main person of this project in the laboratory. and we will have a long collaboration with founder. Thank you for attention.
Thank you, Dr. O.Nureki, for this very interesting and inspiring talk.
Thank you.
And I will abuse my right as a chairperson to ask the first question. So when we will have the most interesting structure, that of Cas9 in complex with non-target DNA. I mean, not non-target as sperm, but with mismatches in the spacer.
Yeah.
Do you or somebody else work on it?
Yes. So mismatch is usually occurred in the latter part of the spacer DNA, and so the former part is recognized by the seed sequence very strictly, so it cannot allow the mismatch. But the rest of the 10 to 8 nucleotides in the DNA spacer, there is no seed sequence, so we cannot check the complementarity. So the Cas9 allow the mismatch and called the off-target activity.
So, but structurally, it has not been investigated so far, right? Especially in the seed.
Yeah. Some other group reported the structure of the off-target mismatched DNA complex. Yeah. Yes. But in this case, mismatched DNA had not Watson-Crick base pair, but a different base pair. But based on the structure, we have many mutations that enhance the fidelity with less off-target activity. So if we incorporate change the arginine residue that recognizes DNA, then the DNA is easily come back to the double-strand DNA. So if you have a mismatch between the guide RNA and the spacer DNA, then the DNA is easily come back to the double-strand DNA to release from the Cas9. So these mutations enhance the or reduce the off-target activity. So based on the structure, we can make such a mutants.
Okay, thank you. Questions, please.
Thank you for excellent talk, Professor.
Thank you.
I mean, I myself, like the structural approach to the modification of the Cas proteins. My first question is, of course, you solved the structure of AS CPF1, but it seems like the common opinion right now is that LB CPF1 is more useful for the editing. Maybe I missed. Is there the structure of similar structure as your AS structure, AS CPF1 structure for LB CPF1 structure available? The crystal structure, I mean.
You mean crystal structure or AS CPF1? Yes.
Yeah, LB CPF1.
LB CPF1.
Yeah.
Yes. We also saw the structure of the LB CPF1, and in the case of AS CPF1, is very strictly recognized TGT PAM. And the three domains strictly pack the PAM duplex. But in the case of the LBCP1, the packing by the three domain is loosened. So it allows the PAM, not only the TGT but or TCT or TC or so such a different PAM recognition.
Okay, is it published?
Yes.
Oh, okay.
In a molecular cell in 2070.
Right, I must have missed it. Another question is related to the deaminases.
Yeah.
So it seems like Cas9 deaminase works pretty well.
Yeah.
The recent report that the LBCPF1 CT deaminase works as well, although with much more narrow window. And we're working on the ABE LBCPF1 fusion and have seen very much less activity and as well very much narrow window. Can you comment? So our interpretation is that the non-target strand is, or basis of non-target strand, it's much less available in the CPF1 complex for the modification than in Cas9 complex. Is that correct interpretation?
Yes. So some group are now reporting the paper the CPF mutant that recognize only TT, not TGT. they produce these mutants by the direct evolution. So we are now analyzing the structure. Man, yeah, we will have the results soon.
Okay, thank you.
Yeah, I'm sure there are much more questions, but we have to move back to the schedule, and I hope Professor O.Nureki will be available for discussions in the breaks. So thank you very much again.
Thank you very much.